Hello and welcome to the session. Here we are going to. A microscope is an optical instrument which is used to produce a magnified image of a given object. So, initially, I'm going to talk about a few basic things pertaining to microscopes, and then later, we are going to talk about different types of microscopic techniques. So, it was back in close to 1600s when people started using microscopes, and it was in 1655 when Robert Hooke he observed a piece of cork under the microscope, and he observed these structures which he called as cells. And then later, many other people they started observing living things under the microscope, and Antony von Leeuwenhoek observed different types of organisms in pond water. One important term pertaining to microscopes is called magnification. The magnification refers to the ratio of the size of the image obtained to the original size of the object. That is how many times the image is going to appear larger as compared to the original size of the object. That is called as magnification. Another important term that is resolution, which is the ability to uh, to distinguish between two regions of a microscopic image. And this is important to show the details in a clear manner. Okay, so microscope these are optical instruments which are meant for producing a magnified image of an object. A simple microscope has only one lens for producing magnification. A common magnifying glass is an example of a simple microscope. On the other hand, a compound microscope contains a combination of lenses to produce magnification. The compound microscope which we use in routine to observe different biological specimens, they contain an objective and an ocular lens to produce magnifications. So here there are two lenses which contribute to magnification. So this is an example of a compound microscope. Light is used as a source to illuminate the specimen for a microscope. So this is called as a typical light microscope. In other cases, we can measure other different type of things. For example, we can measure fluorescence, and then we can use electron beam in case of electron microscopes as well. In order to distinguish the specimen from the background, it is important to have a proper contrast. And many a times, biological specimens are not colored. Or even if they're colored, the specimen is so thin that it is difficult to observe different parts of the specimen. So therefore, we use staining agents to color different parts of the specimen, hence improving the contrast and therefore making it possible to observe the specimen. The refractive index of the material is also an important aspect when using microscopes. Here you can see the construction of a typical light microscope. There is a source of light. A light moves through from this source and it passes through the specimen. When light moves through the specimen is placed on a glass slide, and the cover slip which is used to cover the specimen is also made up of glass. From there, the rays of light they move into air, which is a less dense medium. Glass being a denser medium, so when light moves from a denser medium, which is glass, to air, which is a less dense medium, light bends away from the normal. So this red colored ray of light is showing that the light is going to be deflected away. So all the light rays which are coming from the specimen are not being collected by the objective lens in this case. Here in this example, only one ray of light is shown deflecting here. So only a little amount of the signal in terms of light collected is going to be transferred to the objective lens, and some of the information in form of the refractive light is going to be lost. This is typically important when using higher magnification lenses because the intensity of the signal can significantly decrease because of the reflected light. This problem could be prevented by using a technique which is called as immersion oil technique. In this method, we add a layer of oil, we overlay oil onto the specimen, and the oil is now in a continuous contact with the objective lens of the microscope. So this is shown by means of this blue ray of light, that when light moves through the specimen, this ray of light does not go into air. The thin film of oil produced between the specimen and the objective lens has the same refractive index as that of the glass. Since the refractive index of oil is similar to that of the glass, so therefore bending of light does not take place. So these light rays, the one which is shown in blue color, it does not change its course and therefore is going to move into the objective lens. In this manner, the loss of signal intensity because of refracted rays of light could be eliminated. So this technique or this method is called as immersion oil technique, in which we immerse the lens in an oil which has the same refractive index as that of the glass which is used to hold the specimen. In this manner, because of the same refractive index, loss of light signal does not take place. So now I'm going to talk about a few important techniques which we are going to talk about in some detail in the coming slides. So one of the important technique which we have been routinely using is the typical optical or the light microscope. This method, it uses light rays, and it has a resolution of the order of a few microns. The other type of microscopic technique is called confocal microscopy. In confocal microscopy, it is important to have light coming from only one plane of the specimen. This helps in improving the quality of the image. Electron microscopy, transmission and scanning, they both make use of a beam of electrons. The electrons are incident onto the specimen, and the way the electrons interact with that specimen is used to produce an image. Atomic force microscope uses a probe. The probe physically touches the surface of the specimen, and based upon the way the probe interacts with the specimen, it produces an image of that specimen. So I'm going to talk about some details pertaining to these methods in the coming slides. Again, there is some basic information regarding the units of measurement. Like a micrometers, generally we measure the size of most of the specimen in terms of micrometers, which is 10 to power minus 6 times of a meter. A nanometer is 10 to power minus 9 of a meter. One of the important ways to produce the image of an object is to carry out imaging without using a stain. Using a stain is important in many cases, and it can help in improving the contrast. But sometimes the stains, they are difficult to be applied onto live specimen, and sometimes the stains may react with some of the components as well. So one important thing is to improve the contrast by using staining agents. However, there are other methods in which we can visualize cells without staining. phase contrast microscope, the contrast is produced because of interaction of light rays with one another when their phases change, and the way the light rays they undergo this process of interference that produces a contrast. So this is a picture taken by using a phase contrast microscope shown in this panel. This is a comparison of a phase contrast microscopy with bright field microscopy. So in case of a bright field, you can barely see the cells because the specimen is not stained and the background is also bright. So therefore it is difficult to observe these cells here. As opposed to the bright field microscope, in case of a phase contrast microscope, you can see that the different parts of the specimen are visible versus the background because of the degree of interference taking place between these various reflected rays of light. The next type of microscopic technique is called fluorescence microscopy. This is based upon fluorescence produced by a given specimen. When the electrons are excited, well, in terms of fluorescence microscopy, so there is an exciting laser, and because of that exciting laser, the electrons become excited, and when the electrons they return from the excited state to the ground state, they release energy. That energy is in form of light, and therefore this phenomenon is called as fluorescence. So the fluorescence microscopic technique is used to identify parts of the specimen which are labeled with a fluorescent substance. So only those parts of the specimen which are excited, only they will produce fluorescence and will be detected by the microscope. Different type of agents could be used to produce fluorescence. They are called as fluorochromes. That is one way to produce fluorescence during microscopy. The other method is to use fluorescent proteins. There are different types of fluorescent proteins which are available, and they could be used to color different parts of the specimen. I will briefly talk about fluorescent proteins later on. This is an illustration from the book showing the process of the use of fluorochromes to label a given specimen. The specimen here is a particular type of a bacterium. These green colored triangles are representing antibodies, and these antibodies they are specific for the antigens of this bacterium. The antibodies are labeled with this fluorochrome represented by these yellow circles. So the antibody is conjugated chemically to the fluorochromes, and then this conjugate of antibodies is allowed to interact with the target bacteria. If the target bacteria contain the antigen to which these antibodies are specific, then the antibodies will bind to the antigens, and upon binding of the antibodies, the fluorochromes will also be attached to the surface of this bacterium. If the target bacteria do not contain the appropriate antigen, then these antibodies will not bind. So in this manner, we can use antibodies which are conjugated to a fluorochrome, and we can specifically label different parts of the specimen. And this is a representation of the image obtained through a fluorescent microscope. So you can see that the fluorochrome has labeled the entire surface of these bacteria. As I've said earlier, that different type of fluorescent proteins could also be used to label different parts of the specimen. This is done in the following manner. This is a diagrammatic representation of a plasmid vector. The plasmid vector contains a gene for green fluorescent protein here. Associated with it is the multiple cloning site. So whatever is the gene of interest, that gene of interest is cloned in this multiple cloning site here, and the resulting plasmid is then introduced into the host cells. The host cells will carry out transcription, and the genes, these two genes, the gene of interest and the green fluorescent protein gene, they are placed in such a manner that when transcription occurs, that is going to produce a messenger RNA, one messenger RNA, which is corresponding to both of these genes. When translation of this messenger RNA will take place, it will produce a single polypeptide which will be containing the protein of interest as well as the green fluorescent protein. So in this manner, the cell will produce a hybrid protein which will include the protein of interest conjugated to the green fluorescent protein. So wherever in that cell the protein of interest will be present, it will produce a fluorescent signal because the fluorescent protein is conjugated to the protein of interest. So wherever the protein of interest is localized, at that position in the cell, a fluorescent signal will be obtained. In this illustration, the gene is for the green fluorescent protein. The green fluorescent protein was originally from the jellyfish, but now we know about many other different types of fluorescent proteins in various organisms. So in this illustration, the gene is shown for green fluorescent protein, but now we know about many other different type of fluorescent proteins, including red fluorescent protein, cyan fluorescent protein, yellow fluorescent protein, and so on. So by using different types of fluorescent proteins, we can label a target protein, and then we can find where in the cell that protein is located. This chart shows the absorption and emission spectrum of different types of fluorochromes. So these fluorochromes, they produce fluorescence in different ranges of wavelength, and therefore it corresponds to different colors. So the same specimen could be labeled by using different types of fluorochromes, and upon excitation with a suitable wavelength of light, it is going to produce a particular type of fluorescence. In this manner, if we label different regions of the cell by different fluorochromes, we can obtain individual images showing those different parts of the cell. One important related technique is called as photobleaching. The term photo refers to light, and bleaching is the loss of color. The technique photobleaching is used for several different applications. Many a times it is used to study the migration or mobility of a given molecule of interest. Different type of molecules of interest could be labeled with a particular type of a fluorochrome, and then they could be studied by using this mechanism. It could be used to stain proteins or other type of molecules as well. In this process, what happens is that the fluorochrome is added to a particular molecule of interest. Say, for example, in this particular case, it shows a membrane-bound protein. So the fluorochrome is attached to a membrane-bound protein. Since the protein is present throughout the membrane, so you can see the fluorescence signal coming from the entire membrane. So this green color of the cell is representing that we are getting a green colored signal from the entire surface of the cell. Now, in this process, we carry out photobleaching. So at a selected location of the cell, a laser of a typical wavelength is allowed to be incident, and the laser is going to bleach this area and therefore the fluorescent signal will be lost from this area. In this representation, it is shown by this white color, but actually in a microscopic image, it is going to appear dark. It will be black in color. But in this illustration, it is represented by white color. Now, these molecules which are labeled, if they're mobile in the membrane, they will start to migrate in this region. So there are labeled molecules in the surrounding, and then there are bleached molecules in this area. The bleached molecules, they are going to diffuse away from this area, and the labeled molecules are going to diffuse into this bleached area. This is diagrammatically represented here. So when the bleached molecules, they migrate away, and the labeled molecules, they migrate to this area. So fluorescence in this bleached area is going to be recovered. So this process is called as fluorescence recovery after photobleaching, or abbreviated as FRAP. So this is an important technique which is based upon a fluorescent labeling of molecules, and followed by photobleaching, the fluorescence is allowed to recover in that given area. If the molecules, they are highly mobile, then it will take very little time to recover fluorescence in this area. If the molecules of interest, they are not dynamic, they do not move a lot, then it will take longer time to recover fluorescence intensity in this given area. And if the molecules are immobilized, they cannot move at all, then there will be no recovery of fluorescence in this bleached area. So in this manner, we can study the dynamics of molecules, dynamics of membranes, proteins, and other molecules by carrying out photobleaching. So first, photobleaching is done, and then we look at the process of recovery of fluorescence in that bleached area. Greater is the mobility of molecules, less is the time required for recovery, and vice versa. So this provides information about the dynamics and mobility of the molecules of interest in a cell. In this representation, it shows that the label is present on the membrane. But in many cases, we can carry out labeling of proteins or other molecules which are present in the cytoplasm or in other organelles of the cell. In this way, this method can provide with useful information about the mobility and dynamics of different type of molecules in various compartments and locations of the cell. This is a representation of an experiment taken from a paper. So in this panel, you can see that this is the image taken from a fluorescent microscope, and it is representing the proteins which are concentrated in these regions of the cell. So this oval shaped structure is representing the place where the cell is present, and this dotted circle shows the area which is bleached. So these are the images which are taken before bleaching. Later, when bleaching was done, so you can see that fluorescence has been lost in this region of the cell. After bleaching, as time is increasing, you can see that the proteins from the neighboring area are going to migrate into this region, and later it is going to increase the fluorescence intensity. In this manner, the fluorescence is going to recover. And this is a bright field microscopic image of the same cell. However, in this case, you see that these are the fluorescent foci present on the cell, but when photobleaching was done, this protein was not able to recover in the same amount of time as compared to this one. So here there is quick recovery of fluorescence, and in this case there is almost no recovery of fluorescence. So it shows that the proteins shown in this first case, they are highly mobile. So the proteins, they can migrate in the membrane, they can diffuse and can recover fluorescence in this bleached area, as compared to the one shown later where we do not see any recovery of fluorescence. That means the proteins shown in this second case are almost fixed and they are not very much mobile. Okay, so the next type of microscopic technique is the confocal microscopy. The problem with previously described types of microscopic techniques is that the specimen, although it is a very thin slice, but the specimen contains many planes in it. And when light moves through the specimen, those different planes, they contribute to different rays of light coming from various planes, and all of those light rays coming from different planes of the specimen are being collected by the objective lens. All of those rays of light coming from different planes, they contribute to the image of that object. This many a times is problematic because some planes, they are in focus while other planes, they are out of focus. So therefore it produces a blurring effect on the image of that object. This problem is resolved by collecting light rays only from one particular plane of the specimen. This is done by using a pinhole. Here in this representation, you can see that there are these different light rays in various colors which are coming from the specimen. These light rays are going to be transferred there to the lens of the microscope through a pinhole. The position of the pinhole is adjusted in such a manner that in this illustration only the black ray of light is going to pass through the pinhole. The other rays of light, the red one and the blue one, they are going to be blocked by this pinhole. So that means the light coming from other different planes is blocked, and light coming from only one plane, which is represented by this black ray of light, is allowed to be collected by the lens. And hence these light rays coming from only one plane are going to be collected by the lens and contribute to the image of that object. The position of the pinhole can be changed. Say, for example, if the pinhole is moved upward, now the black ray of light will be blocked and the blue ray of light can pass through the pinhole and will be collected. Or if the pinhole is adjusted to a position lower than this, then the blue and black rays of light will be blocked and the red ray of light will be collected. In this manner, we can look at only one particular plane of the specimen and can produce an image of that object which is going to be much sharper and better, well focused as compared to any other type of microscopic technique. Another important feature of confocal microscopy is that we can produce multiple images of the same object by collecting these light rays coming from different planes. So we can have a 2D image for each of the plane. So what is done is that many images of the specimen are taken corresponding to all these different frames, and collectively all of those images are then compiled by a computer software, and the planes are overlaid onto one another, and this can produce a three-dimensional image of the specimen. So as opposed to the previously described types of microscopic techniques, confocal microscopy can also produce three-dimensional images. So here is the ray diagram for the path of light in a confocal microscope, and this is an image from the book which is obtained by using a confocal microscope. So, since light is being collected from only one particular uh, plane of the specimen, so therefore the image produced is well defined, having good resolution, and is sharper and more focused as compared to the image obtained by using an ordinary microscope. Many a times, the confocal microscopic technique is coupled to fluorescence microscopic technique. That is, we can label the specimen, different parts of the specimen, by using different types of fluorochromes, and then the images could be obtained by using a confocal microscope. This image shows a comparison of different specimen when taken from a regular fluorescence microscope and a confocal microscope. So if you would compare the panels shown on the top with the panels represented at the bottom, you can clearly see the difference in the details which could be obtained by using a confocal microscope. So the images on top, they are relatively blurred because light coming from many planes is contributing towards this image. On the other hand, the images shown in the bottom panel, they are better, well resolved, and of better quality because they represent light coming from only one particular plane of the specimen. In this manner, confocal microscopy is used to produce better quality images, and it could be coupled to fluorescence imaging as well. And by taking images in many different planes of the specimen by using computer assisted softwares, those images could be put together to produce a three-dimensional image of the specimen.